好的，
谢谢。
就是想和大家说一下，我们之后的这个录屏呢，将会分享到我们的 YouTube channel， 并且也会分享到钟老师个人的 YouTube channel。所以之后呢，你们可以通过这些频道去再次回顾一下我们本期的讲座。好，那么现在我们就开始我们的活动吧。大家好，欢迎参加本期 BioCosmo 的学术讲座。我是本次活动的主持人冯源，来自印第安纳大学。首先，非常感谢大家的参与，尤其是美西甚至在国内和日本的朋友们，相信你们的早起和熬夜都会是值得的。在活动正式开始之前呢，请允许我向新来的朋友们再介绍一下我们的社群。我们是 BioCosmo， 面向从事一线研究的海外高校生物学者的实名制公益性社群。主群成员仅限经过身份验证的海外各大高校生物类相关专业的在读博士生及以上学者。我们致力于建立可靠、有爱的社群环境，并通过社群运营团队积极连接外部资源，赋能社群发展。散是满天星，聚为一团火。我们的目标就是凝聚散布在世界的生物之星，激发海外一线华人生物社群的能量，连接优质学术和执发资源，助力生物人的成长，同时为中国生物研究的发展创造价值。目前我们的社群成员已经超过了1500余人。PPT 的右下角呢，就是我们公众号的二维码。陆续我们还会有各种活动上线，所以还没有关注的朋友们现在可以扫码关注。在讲座的最后呢，我们还会附上小助手的二维码，通过添加小助手就可以加入我们的社群。好了，那么接下来就进入我们正式的学术讲座。It is our very pleasure to have invited Professor Sheng Zhong from University of California, San Diego as our distinguished guest speaker today. Dr. Zhong did his undergraduate study in Peking University from 1997 to 2001, majoring in mathematics and economics. After graduation, he went to Harvard University for his PhD training in biostatistics. By using only 3 years, Dr. Zhong finished his PhD study and became officially Dr. Zhong. He then went to Stanford University as a visiting scholar for a year and found a position in University of Illinois, Urbana-Champaign as assistant professor in the Department of Bioengineering. In 2012, he joined the Department of Bioengineering at UCSD and became professor at 2016. He received an NIH Director's Pioneer Award, an NIH Catalyst Award in Diabetes, Endocrinology, and Metabolic Diseases, an NIH Director's New Innovator Award, an NSF Career Award, and an Alfred Sloan Fellowship. He serves as the director for UCSD Center for Liquid Biopsy Research and the organizational hub of the NIH-funded 4D Nucleome Network. He leads the Human Cell Atlas Seed Network and a transformative technology development team for the Human Biomolecular Atlas program. He had published more than 100 papers on high-impact journals and made significant contributions to a lot of fields. Besides scientific research, Dr. Zhong has an incredible mentorship that 8 of his previous trainees are contributing to science on tenure-track faculty positions. And now I'm going to pass to Dr. Zhong to start his talk today. If you have any questions, please type in the chat box and we will pick questions to ask Dr. Zhong in the Q&A session after the presentation. So now, Dr. Zhong, please feel free to take over.
Thank you, Yuan. Thanks a lot for the very kind and generous introduction. 我有2个问题哈，第一个是程序的问题。我看到程序上是说先分享一些经历啊，这个 career route， 然后是一个 more like a presentation and then followed by Q&A， 是这样的吗？
没错，就您怎么喜欢怎么来就行了。
好的，好的，当然可以的。这个还有就是就是在问题啊这些 或者是 career development 这方面的事情，大家觉得讲中文是不是合适？是不是有讲英文的需要？如果没有的话，我就先讲中文，怎么样？
没有问题，没有问题。
好的，好的。好，谢谢这个冯源的介绍哈。这个介绍里面我有一个小的更正啊，这个我并不是3年就读完了 PhD 啊，我是4年PhD 这个在简历上看起来像3年，确实很像，是因为第4年的时候呢，我的这个 PI 的实验室，他这个 Wing-Hong Wang 他从 Harvard 搬到了 Stanford 所以我就跟他搬过去了。那搬过去以后 ，Stanford 当然就是 visiting scholar。所以在我的简历上是在 Harvard 3年 PhD， 然后 visiting scholar， 但是实际上呢，我是第4年结束了以后才回到这个 Harvard 去 defense 这个，所以我有1年 absence 在 Harvard 是这么一个情况。然后
感谢您的纠正，感谢您的纠正。
这个是比较 confusing 的。这个 actually I got advantage on job market because of exposure in a different university。对，所以主持人觉得这些前面20分钟，我看到这个是介绍前面20分钟有一些 career 这些问题，这些是
not necessarily 20分钟，您就简单这样介绍一下，或者是您觉得有什么事情在您的 career 发展上或者是人对您的有巨大影响的事情，随便说一下就好，就不用 exactly 20分钟就。没问题。
可能这个没准对大家可能用处不是一定用处吧，就是可能有不少同学挺关心这个事儿的，就是有一些同学也结果就是因为看到这个 BioCosmo， 然后来跟我交流说想跟我说这个他们可能更关心这一块。那我也就从我自己的 career 里面有2-3件事情，我就开始聊一聊，主要是把这个 topic 聊起来。我也同意，时间是可能如果大家关心，我们就多聊点这个science 的话，这个大家有很多 exposure 了，对不对？这个我们也可以 flexible。对，其实我也收到不少的问题，就是在大学里面我们也有一些活动，有不少同学就是提一些问题，就是可能关心这个对于development， 那对于我的 research direction， 甚至是我的 research direction topic change， 这方面的问题。特别我被问到多的可能是大家说你这个 mathematics 的 undergrad 这个是怎么开始做 biology， 而且我们自己的 lab 里面也有 trainee going both ways， 也有这个undergrad 是 biology， 那现在已经是 tenured professor， 就是 primarily or almost completely on theoretical and computational genetics， 也有就是 both ways， so people go both ways。所以我自己呢是在 undergrad mathematics 并不是我的 choice， 我是被迫的。这个被迫的原因呢，就是 partially because 我在高中的时候，那个时候呢就参加了一个高中的交换学生在美国的 Arizona 的一所高中读了一段时间。在那个时候呢，我的高中96年的时候，那个时候 exchange student 还是比较少见的情况。然后到了 Arizona 以后呢，这个学的这个高中内容就是感兴趣的内容是西班牙语这些，数学他们也不让我学。结果这个再回到中国，马上就是高3了。这个高3一开始就是开始第一次模拟考高考，我们叫1模、2模、3模，3模完了就高考了。就整个高3就是1模、2模、3模高考，我不知道现在是不是这样。我回去基本上还没喘过气来，就一模还是一诊一诊。我就记得我那个数学就已经结束的时候打铃了，我就把数学的卷子的正面做完了，背面完全是空的，还没有翻过去，我都不知道卷子还要翻过去。然后我这个年级第一次诊断性考试，我在年级108名，这个数字也比较好记，跟那个100单八将唐山100单将那个数字一模一样。那这个成绩出来了，这个基本上就是说明这个要想进重点 重点大学的重点线都得努力的这么一个情况。然后当然就使劲努力了，最后还是比较 lucky， 这个就是可以最后还是觉得还是要 fight 一下，这尽量的希望能够去北大清华这样的填这样的志愿。但当时在就是到了第3次诊断性考试，我的成绩可能也才在全年级可能20-30名的情况下吧，那对北大清华来说这还是非常大的 risk。所以那个报志愿的时候，我就想找那个北大清华哪个系收分最低。觉得要是有体育用品保管系，我就填了。有没有？没有。没有的话就填了服从分配，只要是愿意收我的，不管什么系。结果这个考完了以后还是比较幸运，就上了这个录取线。当然也是不奇怪，也就是分配到了数学系啊。在那个时候呢，那个数学系的收分是 among 当时最低的啊，所以也是分配过去的。这是我就是这么进了数学系。但进了数学系以后，有好多的很有趣的事情。就是北大数学系当然是非常的传奇的地方，对吧？这个在我们那一年也是六位奥赛金牌，1位铜牌，7位，这是非常传奇的地方。我就选2件这个在里面可能比较好玩的事情跟大家说哈。一个呢是到了大2的时候开始有选修课了。那个时候选修分2种，一种叫做现选啊，现选其实和必修课差不多啊，就是有一点点的这个选择余地。现选课不管是从这个计分来说，以后保研啊这些都跟必修课是一样的，它是算成保研的分的。另外一种就是选修课，选修课那就是那就没有啥要求的，就是你可选可不选，这个学分也记上，但是这些分以后保研啊，其他的这些这个评优啊、评奖学金对也都不算啊。这个到了大2的时候就开始有第一门选修，完全是选修课，就是数学史。因为是选修课呢，所以它时间安排的也比较有趣，它安排在周6周末早晨7到9点还是8到10点啊。那个时候就是冬天，是一个冬季的，那个时候冬天那个路也不太好，早晨要去就起不来，也要去也比较困难。我记得好像男生这边第1次早晨都去了，选了这个数学史的。第2周似乎就基本上就男生都没去，似乎全部都是女生去了。因为男生到周5晚上那个寝室也不关灯的，就是整一晚上的周6早晨都还刚倒下。到期末的时候，这个时候怎么办呢？要考试，那个考试就是说开卷，大家写一篇论文就完了。我们知道我寝室里面有1位同学，他是坚持去了的，可能男生就1位同学坚持去了。这个正好在我们寝室，我就跟他把笔记本要来，他做了笔记，他做了2本这个大开本的笔记，我就把他的笔记借过来，拿着他的笔记去写了1晚上论文。我先把他笔记先看了一遍，都是些故事，有好多故事，这些里面我就看到一个故事，我当时觉得真的是挺打动我的。那个故事是什么呢？是说非欧式几何是怎么发展起来的？什么是欧式几何呢？就是欧几米德当年写了一个几何原本，里面有5条公理，然后所有的平面几何学全部是基于5条公理的。这5条公理都是特别简单的事，公理嘛，就是过1点可以有一条直线，过2点可以连1条直线且1条直线，这些就是2条公理了。其中第5条公理是不太明显的，他说过直线外的1点可以做1条只有1条这条直线的平行线，这个肯定大家都知道对吧？这个我们中学都学过。但这个事情相对于作为公理来说，就是它不可证实不可证伪的这种绝对的标准正确性来说，它没有其他的那些公理那么显然。你凭什么说过直线外1点只能做1条直线平行线呢？所以这个事情虽然是整个欧式几何的公理，但是呢，在18世纪的时候，就有人希望说挑战这个公理，就开始去发展说其他的可能性。 这些发展的这个文章当然是发表不出来的，当然是被整个学界给 diss 的，对吧？这个当时的就有年轻学者从10几岁，那个时候数学家都是10几岁就是数学家了，就真正的数学家，就是那个时候就从10几岁一直到年老，就是在发展一套系统，就是过平行外一条直线外1点可以做2条平行线的。当然了，这就是始终在一辈子发不出文章的这个数学家，终于可能在他年老还是去世了以后，他的东西被发表出来了。现在是整个非平面几何的基础。那我们在上大学的时候上了1年的几何学，那都是非平面几何，这个就是非欧几何的根基。这个故事，这个我是开始看的是真的是很惨啊，这个是越看越觉得这个，这个学术真的是要想创新这个太难了。这后来我的论文也就是照这个写的，我都记得我的论文题目，这个黑暗中的光明论非欧几何学的产生。然后结果后来这个分数就是那个时候分数都是 announce 的，全年级都看得见。可能现在可能不一样了，我们不让把学生的分都 announce 出来。那个时候结果发生了个什么情况呢？发生了一个我自己觉得很过意不去的情况。就全年级大家的分数呢，都在60到89分之间，有两个学生得了90分，全年级最高分，全年级100多人呢。大家可以想象是哪2个得了90分吧？是我的室友 who absolutely deserves 但是另外一个 absolutely does not deserve 这个 score 的是我，这个我非常过意不去，我也不知道这个情况，我整个就是我就觉得我就把笔记记了1晚上，我并没有想要这样，对吧？这个我是非常过意不去。当时我的这个室友他当然也是确实是肯定是这个受到了一些震动嘛，这个震动当然就后面就有一些影响，就开始我们之间就没有说话了啊。后来我还帮他打开水打了一段时间，这个到了大3以后，这个开始恢复了一些交流。大3的时候呢，这个我已经开始做 research 了。我想这个下面再跟大家聊一下我是怎么开始做到跟 biology 相关的 research。anyways， 大3的时候，我一天我都在那儿做 research， 确实有些课呢也就没怎么去上。这个也有同学问我就是怎么 balance 这个上课啊和这个做 research 的关系，我自己有一些我自己的偏见吧，我一会儿也可以 share。不过 anyways， 我大3的时候，当时我们一门很重要的现选课，说是现选课，其实基本上这个学这个信息学方面的大家全部都选的计算机网络。这门课我基本上也没有去，这说话又到期末了，然后期末考现选一个4分的重课，那我怎么办呢？我当然知道我们寝室谁最用功了，我找他了，舔着脸去吧。这个我说谁，要不然你跟我说一下有没有划重点啊？划了，你画的重点给我看一下呗。把他的书给我，上面画的重点。我很高兴啊，但是你可以想象我们之间的这个时候，他并不愿意跟我多说什么，对吧？这个其中有一个很关键的事情，他都没跟我说，我不知道是他就是不愿意说呢，还是这个太明显了，他觉得不用说。什么事儿呢？就是因为这门课呢，对于数学系的学生来说，还是认为里面有很多内容就是太多、太难、太深了，所以大家去要求开卷考试。这个老师心一软就同意了，这个大3的计算机网络这门课是开卷考试。这件事情全班可能就1个人不知道，就我不知道。我就拿着这个勾了重点的一本书啊，我就熬了几个夜啊，就是我就是要重点的时候，我们的这个 这个可能唯一需要说的一句话就是开卷考试，并没有说，我也不知道。然后那天就去考试了，早晨去了以后，发现哎，我们学院的院长怎么站在这个讲台上？他怎么来监考呢？这个不太应该啊。结果是怎么回事呢？结果这个我们是任课老师答应开卷考试这件事儿被学校知道了，被学院知道了，学院不同意。为什么不同意呢？就是因为现选课是不管是凭这个凭 scholarship 还是以后保研，都是算进去分的，这个考试成绩是算的。那如果有的同学选的那一门现选课，它不是开卷考试，那你以后怎么公平的保研呢？那学院就说这个事儿不行啊，所以学院就 refute 了这个导师的这个任课教师的这个决定。那这个已经是最后的事情了，所以导致考试当天院长站在讲台上说闭卷，书都交上来。然后你们可以想象，就是说这个时候考出来以后，大家可能成绩都不是特别的好。但是呢，导师又任课教师还是很好的，他就是可能是怎么去把分在 curve 一下 scale 一下，大家基本上都及格了。你这个大三，大家也不是说都不努力，你就都不及格也不行，对吧？但因为由于这个分，可能我猜可能是因为这个分 curve 过后，这个时候就出现了这个，据说是这个计算机网络这门课几年来上下几年在数学系没有出现过的一个98分。这你们也可以猜到这个为什么会有人考98分，就是因为只有一个人不知道是开卷考试，就他是背了1周。这个什么是 TCP/IP 啊？什么是两作手啊？这个对，就是有这么些小的经历。然后我这里都讲了29分钟了。OK， 然后简单说了然后我在参与到研究，是因为那个时候李政道先生开始在北大开始设立李政道奖学金，那个是他初始设立的第1年，我正好碰上那一年，那个是可能是98年，我的大2。那个时候李政道先生设立奖学金是希望让这些本科生开始做科研，当时是一个比较前毛的思想，现在大家本科生都参加做科研了。那98年的时候，那个是一个很前毛的想法。李政道说，本科怎么不能做科研呢？我就来设一个奖学金。他这个奖学金呢，是让导师和学生一起申请，导师也得申啊。你这学生这个时候，就有我的本科导师钱敏平先生。我说赵先生他是 female professor， 他就是提说，你可以在是不是可以在基因组里面、基因序列里面去把 transcription factor binding site 找出来？他认为这个事情很重要。在那个时候，那个人类基因组还没测出来呢。这个杨焕明先生回到中国，才开始让中国担任了国际基因组计划的1%，让人类基因组计划1%由中国承担。那个时候才刚刚开始，那个第1台测序仪我还记得是杨先生这个还没有拿到中国的经费支持，自己筹款，自己先拿出自己的存款，然后在当时的这个中科院基因组所让这个职工自己垫钱买的第1台测序仪啊。那个时候当然要分析这些数据，也是是当时的我的导师钱敏平先生提的一个可以的课题。那等我们的毫无门道，都不知道该做什么呢？对，这个是很有幸，我就被选到这个课题，成为这个李政道奖学金的这个一员，然后开始来做研究。这是我开始有一点点这个生物研究，然后后来再逐渐的扩充。我已经讲了30分钟了，大家觉得下面该怎么样？主持人？
非常非常的精彩啊！我觉得咱们可以差不多开始做一个学术的分享了啊！但是非常非常感谢钟老师的小故事，非常的有意思。
好，没问题。好。Okay, so let me try using English so there are requests of this talk so that I feel that when posted on some public channels, there's some other people can get to hear as well. So I wish to speak with you regarding the possibility of making a finger prick test, blood test for Alzheimer's disease. This is not a resolved question. This is
老师，不好意思打断，您现在还在那个 presenter mode。
Okay, thank you. Okay, thank you.
Sorry.
Okay, I'm going to try again. I hope to speak with you regarding whether it's possible to make a finger prick blood test for Alzheimer's disease. My affiliation is with the bioengineering department at UC San Diego. I also would like to disclose that part of this work is done from a startup company that I'm involved called Genemo Inc. Regarding my lab at UCSD, we generally work on the 4 directions. In the upper left corner, I state that we have great interest in develop molecular technologies to interrogate systematically RNA, protein, and genome interactions. We hope to review many interactions among RNA, protein, chromatin at once experimentally. And some of this work now has began its route to be developed into potential therapeutics. On the upper right corner, we work on liquid biopsy, which we will elaborate in this talk. And apparently, liquid biopsy is one of perhaps the emerging new field related to diagnosis of disease. On the lower left corner, we also have interest to develop spatial omics technologies. We hope there's some mechanism that could potentially be reviewed. And the lower right corner, we develop machine learning and AI technologies, not only to provide statistical rigor to many of the analysis of large data set generated from the other 3 quadrants, but also we hope through these approaches, we could potentially review some principles behind all this data. Before going to the specific talk today, I would like to briefly mention about some other quadrants that I won't elaborate. On the upper left relates to molecular interactions. There is a 10-year NIH trans-NIH consortium that wishes to review many types of interactions and generally the organizational structure of the genome as well as other molecules such as proteins and RNAs inside the nucleus. So that this word nucleome relates to nucleus and ome, so everything about nucleus in 4D with 3D as the 3 dimension plus the dimension of time in total of 4D. So this consortium of 4D nucleome aims to understand the principles under nuclear organization, and apparently the technologies relates to genome interaction and RNA genome, protein genome interactions among the most focused developments of the 4D nucleome consortium. Relates to what we've been trying to do on the upper left corner. We hope to develop technologies that would systematically review, for example, all RNA RNA interactions. That would include perhaps microRNA and messenger RNA interactions that we probably are more familiar with, as well as other potential interactions between messenger RNAs, between pseudogene RNAs and messenger RNAs. And a couple of years ago, we have released this Mario mapping RNA interactions in vivo tool to achieve that. Also, that now people have started to recognize that RNA can associate with the genome. So the attachment of RNA on the genome now is regarded as potentially another layer of the epigenome. RNA can modulate gene expression by interacting with the chromatin. So in the recent 5 or 6 years, now that there is a cohort of recent technologies to systematically review which RNA can attach to which part of the genome, that leads to discovery of many new chromatin-associated RNA and start to review their functions. So MARGI, mapping RNA genome interaction, is one of the recent technologies and now seems to be widely adopted and leveraged and to interrogate these type of interactions. And on the lower left corner, so protein-protein interaction is apparently important type of molecular interactions that underline essentially perhaps every aspect of protein function. And we all know how challenging it is to experimentally review novel protein-protein interaction. It can take weeks, if not months, to achieve that for discover one interaction. And so we wanted to provide molecular technologies that would have experimentally review hundreds and thousands of protein-protein interactions from human cells at once. So the proper-seq technique, which I will not dive in depth today, is a way to barcode many diverse proteins by unique DNA, and then convert the question of interrogating protein-protein interaction into DNA sequencing as a readout to read out a protein-protein interaction. So these tools are proper-seq for protein-protein interaction, MARGI for RNA DNA interaction, Mario for RNA interaction are published technologies. And we have an ongoing piece of work that hopefully would allow us to review many RNA binding proteins de novo, as well as their RNA targets. So we would like to call it PRINSIC, protein RNA interaction by sequencing. We hope that can be released on bioRxiv. And with all of this, we hope to integrate all this data to hopefully provide a leap to our understanding of these types of molecular interactions. I also would like to mention about endeavor by several labs, many labs at UC San Diego to initiate this UC San Diego liquid biopsy research center. And so I've worked with our faculty teams at UCSD ADRC Alzheimer Research Center, and as well as UCI Alzheimer Research Center to develop the Alzheimer disease liquid biopsy analysis focus here at the Liquid Biopsy Research Center. We also have a strong interest and research under the direction of breast cancer in reproduction, especially assisted reproduction for human in human clinic, as well as immunotherapy. We have a growing interest in leveraging single cell and also circulating RNA and DNA to interrogate the potential of help to immunotherapy. And so with all of this said, let me converge to one question, not necessary to provide an answer, but provide some discussions to see whether there is a possibility for disease diagnosis using peripheral blood. Before discussing about the current research, I also wanted to mention about some of the lessons learned from the recent 10 years, including the cautions and opportunities that are laid out I would like to do a case study using this company called Theranos as an example. So Theranos was a startup company, but very quickly grew into a giant biotech. At year 2015, not too long ago, it was evaluated as a $10 billion US dollar company. That makes it one of the rhinoceros in the biotech field. And what Theranos does is to develop blood tests that require only very small amount of blood for the test. And the founder was Elizabeth Holmes. She founded Theranos in 2003. And she founded the company partially by dropping out from her college. She was a college student at Stanford University, and she dropped out from Stanford at her age of 19 in order to found Theranos. And one of the reasons that she felt that she could make an impact was because she discovered a pain. When we say a pain in the startup field, we refer to the need, the desperate need from the society. And the desperate need is as follows. A simple blood test would require a punch and a venous blood draw of usually 10 ml of blood. You may have experienced this. Sometimes it's not enough, 2 or 3 draws are required. For some of people who have to do this once in several days, once a week, for months, this could be extremely painful. And I have close relatives who have to do this once a week for several months. And after some time, and there was so much scar on your elbow, and the nurse couldn't even puncture into the vein anymore. You can see that how painful a patient would be to try to get numerous additional punctures and without any success. That would even make great stress to the nurse. So this was a pain to the society, and this was literally a pain to any person who have to experience this. So what Elizabeth felt is that if we can change this to a finger prick, then the pain could be greatly reduced. It means she could only use a tiny amount of blood to revamp how the blood tests are done. And also the market was tremendous. So there are some numbers cited here by MarketWatch.com. And the liquid biopsy market itself very soon is expected to be at $6 billion US annually. So that was sufficient to support many new inventors, including what she believed she could do. The turning point for Theranos was very quick. And I wanted to point out that even though in 2015, the company raised $700 million, it's tremendously successful. 3 years later, the company went down to bankruptcy. The evaluation was 0. Part of the reason was that it wasn't successful in deliver blood tests that would only require very little amount of blood. And the company claimed that it had those technologies. It was discovered that the company didn't, that very quickly the funds were exhausted and the company was closed in 2018. I feel that the knowledge learned partly is that there is need. The demonstration of the need was obvious. I recall that when our lab moved from Illinois to California, one of the things here in California was that in the local pharmacy like Walgreens, we walk into there, we see Theranos machines for blood, for finger prick. You can literally go to your local pharmacy and put your finger there, boom, puncture, and then you should get your result in a few days. So that says how popular it could be if you have that technology. You could change how blood test practice is done. However, whether the technology is improbable, impossible at all, now knowing the failure of that exercise, I wanted to review this whole thing with you together by thinking about what molecules in the blood that potentially can be used for diagnosis purpose. Perhaps everything about the blood has been tried, including pulse, pressure in traditional medicine, and circulating DNA, any cell in the blood, a white blood cell, any kind of white blood cell, any metabolite, metabolites parent important, sugar level, peptide, any circulating proteins. We have people here mentioned exosomes. And one of the molecules that was perhaps much less considered was extracellular RNA. And this is a class of molecules that were poorly tested, poorly investigated. And we wanted to ask if it's possible to measure RNA. And I wanted to give you a few reasons. One of the reasons was that many of the other types of molecular species cells have been heavily pursued already. And at least 3 years ago, those molecules have not advanced into a finger prick level of blood for test. And for extracellular RNA, I wanted to give a very brief introduction. So these are RNA existent outside of the cells, even until the beginning of the year 2000, this type of molecule was not discovered. People didn't know it exists, partly because that very few RNA existed outside of the cell for a good reason. So in the human blood, for example, the extracellular space is filled with RNAs. We need RNAs. People need RNAs because any extracellular RNA most likely would be a virus. So in our blood, outside of the cell, there are so many RNAs that are trying to clean up and digest RNA as much as possible as a defense system for humans. Nevertheless, starting in around the year 2000 and more reviewed more recently, that there are tiny amount of RNA outside of the cell in the human blood in circulation. These RNA tend to be small, tend to be protected. So either because perhaps these are already after RNA's digestion, those are remaining small fragments that somehow not completely digested, or they are protected by exosome or by some other ribonuclear protein. So there are such a species over there. And several years ago, we contributed to quantify in human blood the concentration. We measured it to be approximately 1 nanogram per ml. So that concentration compared to the typical concentration of cellular RNA, this is probably 1,000-fold less. Roughly speaking, the concentration of extracellular RNA in the human blood is 1/1,000 of the concentration of cellular RNA. And these extracellular RNA typically are smaller than full-length messenger RNA. So we measured it to be most likely between 20 to 150 nucleotides in length. Now that there are many other papers out there that the quantified size and concentration, I believe the range is probably generally accepted now. The bottom line is these molecular species do exist. And the question that presented itself to ourselves was that how to effectively measure extracellular RNA from blood, especially from blood serum, and whether that such measurement can be used for diagnosis purpose at all. By saying diagnosis purpose, of course, we mean people, diagnosing people. At that time, we spoke with clinician friends, clinician colleagues, physician colleagues, and we asked what it takes to test another biomarker, a potential biomarker in patient cohorts. We got a very consensus response. That is to say that, well, if you need 10 ml of blood for measurement, you need to take that blood draw from people, and then you need to run a trial. It takes several years. In some of the examples I will show, one of them took 6 years, the other one took 15 years. And you need to draw those samples, and you wouldn't be able to validate any of your new biomarker in the next several years. We said, well, are there already ongoing trials we can participate? They say it's probably unlikely. While there are many ongoing trials, many of them work with blood. However, those blood had their designated measurement. Say they wanted to measure certain protein as a potential biomarker, they need to use that blood. However, if your new test only requires a tiny amount, say microliter level, microliter meaning a drop of blood, that variation is smaller than the variation of the nurse in every blood draw, right? When they draw a whole tube, whether it's several microliter more or less, that is the error zone. That is totally fine. If most of the trials, they have the ability to spare microliter level, tiny amount of blood for another measurement, if the other measurement only require a very tiny amount of blood to measure. Knowing that, Genemo, our startup, in collaboration with UCSD, we started to try to develop a technique that potentially only require 1 drop of blood for measuring, for sequencing extracellular RNA. And I wanted to mention 2 of our elite collaborators, Dr. Shu Chen here at UCSD and Dr. Irene Su here at UCSD Morris Cancer Center, that we collaborated in this paper to demonstrate from breast cancer recurrence patients how literally from 1 drop of blood, you can have some way to distinguish, to predict which of the chemotherapy treated people would potentially develop recurrence of cancer. And let me go back to the principle. So we wanted to try, and we thought it's possible, and it's not completely impossible, because we knew at that time the concentration of extracellular RNA is 1,000-fold smaller than the cellular RNA. But still, at around the year 2015, it was already 6 years after the initial publication of the first publication of single-cell RNA sequencing. So at that time, single-cell RNA sequencing was already becoming a reliable technology. We knew, people know that we can reliably sequence RNA from a single cell. The total amount of RNA from a single cell is about the total amount of 1 drop of blood serum. The reason is that the volume of 1 drop of blood serum, say 30 microliter, that is about 1,000-fold larger than the volume of a cell. Now, the total amount of RNA in 1 drop of blood, in theory, is comparable in the order of magnitude to 1 drop to 1 cell. So from a back of envelope calculation, Josh Zhou, who I will mention in a minute, at Genemo, thought that there's not a physical barrier between the difference. If there's a similar amount of RNA there, possibly there could be a technology. Having that said, it still took us several years to optimize that, to make it an actual technology. So we delivered this technology to the community, which we call small input liquid volume extracellular RNA sequencing, SilverSeq in short. So what SilverSeq does is that it only requires the input of 5 microliter of serum. That is only like 1/7 of a drop of blood. So here is an example that I wanted to demonstrate and to show the quality of RNA sequencing library from one drop of blood serum. So we could split it into 8 aliquots. Here I didn't split it into the same volume. We had the first 2 aliquots of 3 microliter and 2 aliquots of 5 microliter, 6 microliter, and 7 microliter per se. And here are the size distribution of the final sequencing library as a quality check to see if we could generate a good size distribution that is suitable for a typical Illumina sequencing platform. And I wanted to very quickly to jump to illustration through this clinicaltrial.gov registered trial run by Dr. Irene Su. It's called a POISE trial, short for Predictors of Ovarian Insufficiency in Young Breast Cancer Patients, where she followed chemotherapy-treated young breast cancer patients for 6 years. And among that 5 to 6 year follow-up, some people had recurrence of cancer, very unfortunately. Some didn't have recurrence. And still pending unsolved clinical challenge is there any molecular biomarker that potentially can help to predict who may have recurrence? That apparently has a huge impact to how to manage the patients, how to potentially watch and treat them. And in this particular follow-up, there were 28 blood samples from people that are from people with recurrence in this window of follow-up. There were 68 samples from women who didn't have recurrence in this 5-year follow-up. And we were able to measure extracellular RNA by sequencing using 1 drop of blood, literally speaking, for the same reason as I mentioned before. And most of the blood were used for the original purpose of the original trial. In this extracellular RNA profile, turned out that we could use cross-validation to demonstrate that you could actually have a fairly accurate prediction of who would have recurrence of breast cancer through their blood. So I wanted to quickly summarize. So extracellular RNAs are different from intracellular RNA in size and concentration. Now, SilverSeq technology can sequence extracellular RNA, and extracellular RNA sequencing can be used to classify the blood serum samples from patients with and without cancer recurrence. I mentioned that this work was led by 2 scientists at that time at Genemo, Josh Zhou, and at that time, a visiting PhD student to my lab and intern at Genemo, Dr. Qiuyang Wu. Now that Qiuyang is Dr. Qiuyang Wu by those 2 young scientists. So they worked together around the year 2017, 2018, 2019. They published their work jointly as co-first authors in 2019. And that was 3 years ago. 3 years later, in March of 2022, and I was fortunate to join their wedding here at San Diego with 2 of our lead authors at the center, and also 2 of our other co-authors, now Dr. Yuan Liu and Dr. Zhi Jieqi here for a science bounded couple. And with that, I wanted to get into Alzheimer's. 1 in 9 people who are of age 65 or older are living with Alzheimer's disease. Unfortunately, 2/3 of these people who live with Alzheimer's disease are women. In 2060, by the time that the audience here become elderly, it is projected that 14 million people will be living with Alzheimer's disease. How many of those people is? It is twice of the total population of the Washington state. You imagine that there's 2 Washington states, entire population of people will be living with Alzheimer's disease by the time that you are at the age of elderly. A very brief overview of some of the symptoms and pathology of this unfortunate disease. People with Alzheimer's start to be confused with where they are and what time it is. They start to forget what year it is, whether they are in US or in China. They don't know who's the president. They start to demonstrate poor judgment. You see some of the people start to speak publicly for things that no one should speak in that way. And they start to be forgetful. They forget where they just came from. They have trouble in repeating simple sentences who were just told. And in a very brief pathology, neuron death is vastly observed. Brain size shrink, and this called beta amyloid plaque start to form. You can actually visualize them in human brain. Unfortunately, treatment methods are extremely limited. You may have seen recent news. There was one newly approved drug in 20 years. Unfortunately, that newly approved drug didn't demonstrate good response either even after it's approved. So for more than 20 years now, there haven't been a substantial development on the treatment front. That probably contributes to why the projected number of Alzheimer's people rise so quickly, because there's essentially no way to effectively treat them. Then what should we do? I would like to cite from Alzheimer's Foundation, Alzheimer's Association, of is there any potential way to consider how to potentially make a change? I would like to quote, "What if we could diagnose Alzheimer's disease before symptoms started? The hope is future treatments could then target the disease in its earliest stages before irreversible brain damage or mental decline has occurred." So that has become the main thought of what to do scientifically from here on. How to detect it early? Brain is a very special tissue. Tissue biopsy, while popular perhaps for most other tissues, is nearly impossible for brain because the damage is obvious. You literally cannot take a piece of brain unless this is horribly devastating. Usually, if this is not a brain tumor, it is very rare to do tissue biopsy. Without tissue biopsy, you do not have cellular or molecular measurements of the organ or the tissue that is potentially have a problem. And so that speaks to how diagnosis is very limited. You pretty much are limited to the symptoms rather than having an objective measurement. So almost the only thing that's left to try is blood. So the question becomes obvious. Is it possible to develop a blood test for Alzheimer's? And to us, it's whether it's possible to develop the test based on extracellular RNA. Other molecules are being tried at this moment where we particularly would wanted to know whether the extracellular RNA represent any of the gene expression of the brain. If so, is that a venue or a bridge that we can leverage to learn something about the progression of the disease from the brain? There we were fortunate enough, partly because we can use very little blood to work with at that time ongoing trial at UC San Diego led by Dr. Edward Kuhl. So this was a 15-year follow-up study of people who were recruited from year 2000 to year 2015. And the people were recruited to the study when they were of age 65 or older. And when they were recruited, they were clinically diagnosed. If they didn't behave or have any Alzheimer's symptoms, they are classified as controls. And if they were already diagnosed when they entered the study, they are classified as AD in the next follow rows. And there were a group of people when they originally were enrolled, they were diagnosed as normal. But during the follow-up, at some time after several years, the diagnosis changed, right? They became older. So they started to have either dementia or diagnosis into Alzheimer's disease. So mild cognitive dementia is usually an earlier diagnosis with a smaller, less symptoms, but they could further develop into Alzheimer's. And so we have this group of people who were normal to begin with, but as time goes by, they change into disease diagnosis. So here we identified 35 participants. We identified by that after their deaths, their brain pathology, this is analysis of the actual brain, could actually confirm whether these people are normal or Alzheimer's disease. So their diagnoses are confirmed by pathology. And throughout their enrollment, their blood were collected at the particular time. So the graph on the right shows that every row is a person and every dot is a blood collection time. So you can see that some of the people, the blood was collected multiple times, and in total, there were 164 blood plasma samples were collected. And before we interrogate about the sequenced extracellular RNA in this blood, we also wanted to know what happens to people's brain. In fact, that in the past 10 years, there were several large cohorts trying to generate gene expression profiles from Alzheimer's brain and compare to control brain. Here, I would like to highlight 3 cohorts that are done by Mayo Clinic, Mount Sinai, and Rosemap. I named those 3 because those 3 are NIA National Institute of Aging funded cohorts. And in these cohorts, they used RNA sequencing on several brain regions that are related to the Alzheimer pathology in approximately 80 to 100 plus donors with or without AD. And they carried out RNA sequencing. And it's actually new, even before those cohorts, there were very few genes at gene expression level. By gene expression level, I mean RNA abundancy or protein abundancy level, not post-translational modification, but just about abundancy of the RNA or the protein level. Very few genes are consistently up or down regulated in AD. And this was actually confirmed when we analyzed these 3 cohorts together. From the 3 cohorts, we wanted to identify a subset of genes that are consistently upregulated or become higher expressed in AD. So we had our own requirement. This is not the only possible way you can analyze this data, but with our requirement, we require certain fold change and statistical significance to call a gene to be higher in AD than normal, and then we need it to be showing up in at least 5 of the brain regions in at least 2 cohorts. Altogether, there were only 28 genes that satisfied this criteria. Also, that confirms the idea that not many genes can be reproducibly shown to expressed at a higher level in AD versus control brain. And we wanted to start from those 28 genes to see how do these genes behave in the blood? How do their extracellular RNA behave in the blood? In the left, I would like to to show all the genes with respect to their extracellular RNA level between AD donors and control donors in our cohort. So the lowest distribution of all genes show t-test with positive being extracellular RNA of this gene is higher in AD than control in our cohort. And what we see is in general that there is perhaps not a clear bias to higher or lower. Some genes express higher and lower. That conforms to what would you expect from a distribution. I wanted to bring you to the top level. The top level are the genes with expression increase in AD brains, meaning the 28 we identified in the previous slide. Those are the genes that in previous cohorts to show some consistency in upregulation in AD. Now, if we plot them into with our blood serum, blood plasma extracellular RNA level, you see that most of them are still centered at 0, but a small population shows up to be high in AD. Perhaps suggests that a subset of these consistently upregulated genes in brain have some consistent upregulation in their extracellular RNA level in blood. I wanted to show this again on the right panel. On the right panel, every column is one of the 28 genes that were defined as consistently upregulated in AD brain. And on the y-axis is a comparison between AD and the control blood. So a positive value means that this gene not only exhibited consistent upregulation in brain, but also in our data shows up level, higher level in extracellular space in blood. And what we see is that more of the genes are higher in blood than smaller amount of genes are lower in AD in blood, suggesting that there's still some consistency between what you see in blood and what you see in brain, which is unusual because otherwise we don't really know whether the blood reflects anything of the brain. And among these, apparently the gene on the right would be a potential candidate because in blood, again, it shows a good separation between AD and control. So I wanted to expand that for you and to see how it behaves. So if we expand this gene and on this panel C, this is a plot of their this gene PHGDH expression level in every cohort and every analyzed brain region that related to Alzheimer's disease. So the box plot in red are the distribution in AD brain, whereas the box plot in blue is the distribution in the control brain. You see that this gene is consistently upregulated in AD in nearly every cohort analyzed and every AD-related brain region analyzed. These were from 3 previous cohorts. And in our cohort here in panel D, you see that the extracellular RNA level is consistently higher in blood than control. That says that potentially PHGDH may be a biomarker. I wanted to quickly summarize here. PHGDH is consistently upregulated in AD brains in 3 cohorts, and PHGDH genes extracellular RNA is higher in AD patients' blood than control. So the almost obvious immediate question is what cell types in the brain express PHGDH, and how does PHGDH expression change in each cell type? Does the PHGDH protein level increase in AD hippocampus in brain? Hippocampus, Haimati, is potentially the most critical brain region to the disease. Does the protein change? How does the protein change in hippocampal, human hippocampus in AD would be a critical next question to potentially start to understand why there may be a consistent gene expression that now we observed in multiple cohorts. I wanted to start with the first question: what cell types in brain that express this gene? And with this question, we reanalyzed a single-cell RNA sequencing dataset released relatively recently in 2019 that generated a single-cell RNA sequencing from 24, 15, and 9 people with no AD, early AD, and late AD. Here we are replotting the expression level of PHGDH in 7 cell types in human brain. The reason we can get cell type information is because now we have single cell expression data, and using that single cell expression data, we can have assessment of what cell type it is. So here on the columns, we have astrocytes, endothelial cell, excitatory neuron, inhibitory neuron, microglia, oligodendrocyte precursors. Those represent perhaps the main cell type you would expect in the brain. And we have 3 columns here in 3 colors that represent the single cells from people with no AD, those are control, people with early stages of Alzheimer and late stages of Alzheimer in green and in blue and red respectively. And you see this is the single cell expression level of PHGDH. What we see here is that PHGDH is expressed in astrocyte, oligodendrocyte precursor, not in every cell type. In astrocyte per se, you see that from no to early to late Alzheimer's, there is a stepwise progression of the expression level. So now we started to see what cell types express this gene, and also the change of expression level of this gene actually starts from the early stage of the disease. And I promised to go into a human hippocampus. So here at UCSD, we were able to carry out a study of a 21 postmodern brain. We immunostained this protein in their hippocampuses, including 10 control and 11 AD brains. So here is an example. On the right, you see immunostaining of this protein in the AD brain in hippocampus. You see this staining of the protein that appears to be obvious as composed to the staining of the PHGDH protein in the control brain that doesn't seem to stain as clearly. So if we ask a pathologist's opinion about what cells are stained, so these cells you can see that seem to have a star shape are mostly astrocytes. So those are supporting cells to work with the neuron, and they seem to express a high level of the protein, especially when the AD get progressed. You can quantify this. So these start to demonstrate that something you measure from different patient cohorts, even from the blood, actually indeed reflect some changes from the brain and indeed relate to the correct brain location and correct cell type in the diseased brain. And finally, there is an open challenge in the field is, while this person is alive, right? That's most important, is before you get the postmortem brain. Is there any molecule that you can measure that potentially correlate to this person's cognitive ability? Meaning, do you have any objective measurement that can help to diagnose about the person's cognitive ability? And cognitive ability currently is measured by, you essentially have a standardized test, like you ask the person to carry out some tasks, to answer some questions, repeat some sentences, and then that's quantified, right? And so one of those quantification is using a task that is called a dementia rating scale, and eventually give a quantitative score. The larger the score it is, by answering those questions, it means that clinically it's interpreted as a better cognition. A person with dementia or Alzheimer's disease usually would reflect it as having a lower score. Now, at least for those people that we have got the brain, we can quantify their PHGDH protein level in their hippocampus. It turns out that these two measurements have a strong correlation. It's a negative correlation. The higher the molecular level of PHGDH it is in the person's hippocampus, the poorer the person's cognition ability it is. Despite that, I want to acknowledge that this is so far a small size study. Only a small number of people who had this test in our clinic, and we had their postmortem brain. But still, now that we seem to have an indication, there is a possibility of a connection between a molecular measurement and a person's behavior, a person's cognitive ability. And so with that, I want to summarize PHGDH mRNA exhibits a sequential increase in brain astrocytes as AD progresses. PHGDH protein level is high in Alzheimer's hippocampus, and hippocampal PHGDH level negatively correlates with the person's cognitive ability. I wanted to mention that this is led by my colleague, Assistant Professor Dr. Xu Chen here at UC San Diego, and Riccardo Canandrelli, a scientist in my lab, to make these observations. At the very last, I wanted to go back to the question, can plasma PHGDH extracellular RNA level predict AD before clinic diagnosis? After all, that was our hope. Is there any way to diagnose it early? Well, the person apparently is alive. You can't get to the brain. Can you have a quantitative way to identify the disease even before the symptoms develop? In fact, there is a group of converters whose data we have not used at all, right? We mentioned that in our 15-year follow-up, there were people who were normal, however, unfortunately converted to AD during the follow-up. We didn't interrogate this set of data at all, right, so far. So if I were to plot their PHGDH extracellular RNA level one by one, now in every panel is a person. It's converter 1, converter 2, and so on. If I were to plot their plasma PHGDH extracellular RNA level on the y-axis and their follow-up year on the x-axis, then we have everyone's data in one panel. So we reordered time zero, year zero as a year their earliest clinical diagnosis of the disease was found. So before they were diagnosed as Alzheimer's were the years that now we label them as -1, -2, -3, and so on. After the first diagnosis into a disease, the years are labeled now as positive years. This way, so we recentered the first time of diagnosis as year 0 so that it's more comparable. And from this data, you can see that the PHGDH extracellular RNA increase as the person progress into clinically recognized diagnosis. So we can see this in this summary plot. So every dot in this summary plot is the slope of the fitted curve across time. So now that we have reduced the entire collection of longitudinal data of a person of this 15-year follow-up into one number, that number is a slope. The higher the slope, the larger the increase it is across the follow-up. So the first group in blue are the slopes of our control. The green, the last group, are the slopes of the converters. You see that there is a populational difference. So in controls, the slopes are almost near 0. Most of them are near 0, meaning that while these people are old and become older, as long as they do not develop into Alzheimer's pathology, the PHGDH level in their blood seems stable. However, if they convert into AD, there is usually a spike of increase of their PHGH level in their blood. So I wanted to quickly summarize. PHGH extracellular RNA level increases before AD symptoms develop, and this data nominate PHGH extracellular RNA as a biomarker for early diagnosis of AD. So this work was led by Dr. Zhangming Yan, at that time a postdoc, now a lead scientist in Novo Nordisk. With that, I wanted to quickly mention some aftermath. So there are patient advocate groups who are very much eager to learn about any new potentials in any development on either diagnosis or therapeutic fronts. So being patient is one of those venues. They heavily reached out to us and mentioned about the possibility of early diagnosis may allow for early treatment. What does PHGDH do? What is the potential mechanism? Why it could serve as a biomarker? So the known function of this phosphoglycerate dehydrogenase PHGDH gene is an enzyme. This is an enzyme essential for serine synthesis, especially for the brain. So in the glucose metabolism pathway, glucose can be converted into the following two molecules. Either it could be converted into pyruvate, or it could be converted through a branching route into serine and then glycine. So PHGDH is actually a controlling enzyme to the route from glucose to serine. So with PHGDH, you could brain could produce serine and glycine in the brain, which are essential nutrients. So if we were to review about this known mechanism of PHGDH, how does this known mechanism connect to the current knowledge of potential causes of AD? As one of the highly hypothesized cause of AD is so-called excitotoxicity. That is firing of neuron all the time, and it essentially makes the neuron too tired and exhausted, and finally causing neuronal death. So the neuron has become excited all the time and essentially become toxic, excitotoxicity. That requires the calcium to be released from presynaptic to postsynaptic neuron through for the excitation of the postsynaptic neuron. The calcium goes into a channel. So the membrane channel, one of the perhaps most important membrane channel is so-called NMDA channel, receptor channel. This channel would be closed most of the time, but when it opens, the calcium comes in. It opens when two ligands are bound. One is glutamate, shown in red. The other is serine or glycine, shown in blue. So you see that this is like a gate with two keys. You require the two keys to be both turned open to open the gate. And one of the two keys is synthesized by PHGDH. So we hypothesize that if you have a high level of PHGDH in the elderly's brain, you make this control system too often prone to open, because previously you need to satisfy two criteria. You know that glutamate is often abundant, especially in glutamate neurons. Now that you make the other key more likely to be at the open state, that potentially potentiates excitation all the time, that leads to excitotoxicity. So with that, I wanted to mention a part of our team, including I mentioned Dr. Edward Koo, Xu Chen. I would like to acknowledge our pathology professor Annie Henniker, Douglas Glasgow on the clinic front, and many of our other collaborators and many of the team members I mentioned in the talk, but not all of them. I hope to acknowledge every of them. And finally, I wish to mention an anecdote. So if we Google serine, this essential amino acid, we would find many ads of this supplement. In fact, for me, even before I Google, perhaps AI was already intelligent enough to already feed me serine and ask, are you going to buy serine? Probably because I analyzed serine related research, and somehow AI believed that I wanted to buy this supplement. So this supplement is touted to be a brain booster, to boost your brain. Let me first say, if I wanted to argue this supplement has any use, what would I say? If it has any use, I feel that this probably has some benefit to the children with a mutation of PHGH gene. The people with a mutation of PHGH gene actually would develop a familial disease. So this is sometimes called a PHGH deficiency or serum deficiency, because PHGH is essential for making serum, and serum is an essential nutrient for brain development. You can imagine that if there's a mutation in this gene, the young brain would not have sufficient serum and it would not develop fully. So that people with mutations of this gene often develop this phenotype called a microcephaly, and that means small brain. That is very unfortunate. And you can imagine that the children with microcephaly, because that they do not have sufficient serum, and a serum supplement could be potentially helpful to these children. Nevertheless, by now you probably feel that these ads are not targeting this small population, right? It's really wanted to sell, perhaps want to sell to a large population, including like boost your brain. Most of the time, these are sold to adults. Would serum supplement help adults? Turned out that most cohorts failed to confirm any decrease in serum levels in the brain or cerebral fluid from adults. So serum levels were not decreased in AD. And our study also confirmed that PHGDH didn't decrease, but increased in AD's patients' brain and blood. So therefore, this supplement may not be as helpful as it may claim. And you may know that this supplement has its own challenges that, for example, we mentioned a candidate mechanism for it to overactivate neuron activity and essentially potentially could lead to excitotoxicity, but serum also has other effects to lead to progression of cancer. And so our study got very quickly took up by professional societies and neurologists. So there are, for example, ASMB quickly warned about this supplement, and the KPBS public radio station in its evening news also made this news.
Researchers at UC San Diego have identified a gene marker for Alzheimer's disease. KPBS Science and Technology reporter Thomas Fudge says this means we can better predict Alzheimer's and maybe find the path to a cure.
The study looks back on 15 years of blood testing and observation of hundreds of people over 65. And researchers say the result is an undeniable connection between an overactive gene and the development of Alzheimer's disease. UCSD bioengineering professor Sheng Zhang says their findings were a surprise. That's because the gene and serine, the nutrient it produces, play an important part in brain development.
It's like an overshoot of the good guy, of the good stuff that seems to mark unfortunate disease outcome.
The study tracked the development of Alzheimer's over time in people who were symptomatic and asymptomatic. The expression of the overactive gene, that goes by the acronym PHGDH, shows up as these dark star-shaped cells, a strong contrast to the same cells in a healthy brain. So far, Zhang says they have proven a correlation between the active gene that produces serine and Alzheimer's, and while they haven't proven cause and effect, they think that's what's happening.
Our hypothesis is that a prolonged oversupply of serine makes neurons too active for too long a time and thus become toxic,
which can lead to the conditions that lead to Alzheimer's. There is no cure for Alzheimer's disease, and the UCSD researchers have not discovered one. But Zhang says there are ongoing experiments on mice and on brain samples that could lead to a way to tone down the overactive gene and the overproduction of serine.
To either ameliorate the development of the disease or even reverse the development of the disease. Those are the tests that we are currently doing.
Ironically, some businesses sell serine as a dietary supplement to increase brain function. Based on what he's learned, Zhang says for those who may suffer Alzheimer's, those supplements may be just what they don't need. Thomas Fudge, KPBS News.
So the take-home message I got from this KPBS Evening News is that while the team members do all the work, the PI gets onto the TV. The other good note is that so this ad, Boost Your Brain, previously was on Google and Amazon. So now that somehow it's been taken off. So it looks like that there's the society as a whole, the community as a whole may have taken some advantage of the warning that we were able to provide through our research. So with that, I would like to thank everyone here with me. Also, I want to open for questions. I already see many questions in the chat. I can read through them, or the people who are still here, perhaps you can first speak up to ask your questions. And even if you have posted on chat, then I can go back to the chat to finish the other questions. How about that?
So thank you so much, Dr. Zhong, for your amazing talk. I want to propose a round of applause for your talk first. And here goes our Q&A session. So I will ask questions by the order they showing up in my screen. So the first question, yeah, I want to take the advantage as a host. I want to ask my own question first. So,
please.
So PHGDH, the protein has a role in the pathogenesis of AD, but I'm wondering, does the exRNA of PHGDH itself has any biological function, or it's just a marker?
好，这个非常非常重要的问题。第一呢，这个我想说，这个它的 PHGDH 在这个病原上产生病的这个过程当中，这个病的 etiology 上，这件事情是以前不知道的啊。在我们2020年的第一篇 Human Cohort 严章明et al. 之前 ，PHGDH 不光没有被作为一个病理病原学的进来研究，至少对于 Alzheimer， 甚至在这个各种 association study、GWAS study 都没有 identify PHDH as a gene correlated with AD。如果我们去一些 database 去找这些各种基因，由于各种各样的原因和 AD 产生了任何关系的话，现在可能已经多达2-3千种 genes 了。这个 genes 在这么广的一个范围里面 ，PHDH 都不是 one of them，at least before year 2020。但是最近2年就不一样了。这个前2天我们同学去查这个 PHD 在 Wikipedia 里面。结果这个 Wikipedia 也 cite 我们的工作，说这个它 PHD 是一个 AD biomarker。不过 anyways 早在就是最近在两年以前就都没有完全没有这个进在这个 scope 里面。现在我们猜测这里可能是有原因的。这个原因是什么呢？因为就是说对于好多病吧，包括 AD， 我们怎么去找致病基因呢？用这个 genetic association GWAS 的方法还是最 popular 的，对吧？我把有病的人的基因组测出来，没病的人基因组测出来，他们的主要区别在哪儿呢？最强的区别在哪儿呢？这个 GWAS 这套方法还是一个从 genetics 到 mechanism 的可能一个主要的 approach。特别是对于 AD 这样的 disease， 我们还啥都不知道的，至少特别少的这种情况下，这种 approach 是可能是最重要的。这些 approach 会找到那些 genes， 这些 genes 尽管 mutate 了，但是人仍然可以活到老的，因为只有这些人还活着，他才能测，对不对？但是 PHGDH 这个 gene 呢，它首先它是一个 essential gene， 它对脑的发育是必须的。你看，如果它 mutate 的话，就 microcephaly 这些人经常是没有办法 develop 到老年的，对吧？他们早就出了很多的 unfortunate 的问题了，甚至都可能不能 develop 成成人。那这些人的话，他们其实是不被包含在 GWAS study 里面的，对吧？就是我们的 genetic study 里面研究老，所以这个 gene 呢，它是太重要了，它不能两边都不能出问题，它太少了不行。我们猜测它在老年以后是太多了也不行，也有太多的问题。这个呢，就让我想起来，就是说，这个从研究的思路上让我一直想起来一个事情。以前一直有一个故事，说这个2战的时候呢，那个是这个盟军吧，想要加固飞机，他想加固飞机，我铁板加在飞机的哪儿呢？那就是我们做研究吧，这个飞回来的飞机看看它的那个枪眼都在哪儿。枪眼这个发现好多都在翅膀上，这个说那应该加固翅膀。后来有人说不对，可能打到发动机的那些呢，那飞机都没飞回来，所以你这个分析飞回来的这个枪眼都在哪儿呢？这个不一定是对的。就这个 story 一直让我这个 curious about 我们现在就是用 GWAS 去，尽管这个仍然是有很多很有用的 findings， 而且还是很多 findings 已经开始 translate to function。不过我仍然还是 curious， 因为 GWAS 毕竟是这些已经活到老的人呢，就是飞回来的飞机里面去找的最 popular 的 pattern 嘛，这个是不是有一些这个大家压根就没飞回来，也是很重要的呢？这个是基于这个猜测，是我们为什么觉得这个像这种就是 gene expression level change， 就是 epigenetic 的东西啊，它仍然可能会提供很有用的信息，尽管在 genetics 上我们没有看到它是有的原因。冯源的第二个问题是这个 RNA 本身有没有 signal 的作用？首先这套方向结果是在最近几年被 NIH 定为一个 NIH 的这个重要的发展方向，被 NIH Common Fund 定为重要的发展方向。NIH Common Fund 是这个NIH 的 Director Office 定的这个全 NIH 的方向，它不局限于哪一个 institute。那这个呢，他们在 NIH 就成立了一个 consortium， 叫做 Extracellular RNA Communications Consortium。他们就是来说这个事情还太新了，他不是说我哪一个 institute 已经囊括了这个东西，对吧？这样我可以支持 R01， 因为这个毕竟那些 research 是什么方向大家都理解，但这些刚刚出来的方向还不能理解的，这个一般的 review panel 都没法评的这些东西，对吧？这个怎么办呢？这个就得靠 这个 director's office try to 这个 make 一些audacious 的前瞻性的这个步伐。这个我刚才介绍的 4D nucleon 4-dimensional nucleon 就是 one of them， 这个是一个 common fund project， 就是说这个是一个前瞻性的。这个 ERC extracellular RNA communications consortium is one of them common fund project transnage， 它就是来问有没有这些它有没有 signaling 的功能，它是不是要来 communicate。在 Alzheimer 的 PHDH 是不是有 signal 的功能，我们还在研究不过在有一些cancer 里面有一些 extracellular RNA， 它开始去 signal 其他的 tissue， 去在远处制造合适它的microenvironment， 或者它 signal一些其他的 organ 在去 provide 更多的，比如说 hormone 或者 insulin， 或者是更少的这些远程的利用 extracellular RNA 开始做 signal 的，现在已经有一些很 exciting 的 frontier 工作了。我们自己的 lab 呢，我们也有一些工作，它讲的不是脱离 cell 的 extracellular RNA。我现在今天我讲的 talk 主要是指这些脱离 cell 的 extracellular RNA。我们最近就是诺曼黄的 thesis 的工作，我们发现呢，在一些细胞的表面呢，它在细胞的表面的外面会有一些 RNA。这件事情呢，就好像说以前凡是细胞表面的 molecule 的发现，都是开辟了一个 field。这个表面的 receptor 是吧？这个 glycans， 这个甚至不是开辟一个 field， 这个一类 receptor 就开辟了一个 field。这个GPCR itself 就是一个 field。所以为什么这个东西这些都是开辟 field？ 因为到底有哪一些种细胞？那个表面嘛，就是脸嘛，对吧？这个细胞自己说我是谁，靠脸嘛，互相 communication 看脸嘛，表面是什么就是脸嘛。所以表面有什么，那就是脸上有哪些五官，有哪些眉毛了。但是如果有一类新的 molecule as a molecular class， 它就在表面上，我们以前都不知道，就是有可能细胞表面长了头发，我们都还不知道呢，对吧？所以我们也 contribute 到这个 revealing 这个 cell surface RNA exist and modulate cell-cell communication。这个我似乎也是一个有可能的一个 exciting frontier。它不是 PHDH RNA per se 是另外我们这个最开始的 proof of principle 是monocyte 和 endothelial cell 之间的，这个 monocyte 得粘到 endothelial， 然后再穿过去变成 macrophage。这个过程结果 to some extent 是要用到cell surface RNA 的。这里我们的猜测，这个才是冰山的一角的一角。这个有可能这个细胞用 extracellular RNA 来做一种这个 self identity 的 presentation 或者是 communication 的这种这个可能是 much more popular than the first example we demonstrated。谢谢。
Thank you so much。谢谢，非常感谢钟老师。那么现在因为时间有限，我尽量的更多的呢问大家提出来问题。我先从这个 chat box 里面的问题来提。举手的朋友们呢，可能稍等一下，看我们是否有时间。好，首先第一个问题 from 彭飞。The question is, wonderful talk. I'm wondering how we can enrich the extracellular RNA specifically from the high background of cellular RNA in severe sick. Thank you.
对，这个首先呢，这是个很好的问题，这个是个很重要的问题。首先就是我们 demonstrate 在 patient 里面的血这些都是血浆或者是血清，这些我们并没有全血。这个原因还是很简单，全血是一般来说，这个 clinics 是不储存全血的所以他们拿到血以后就已经 try to remove cell from it， 所以只有 plasma 或者是 serum 会 usually 会 typically get stored。当然我们还会做一些 validation， 我们会做 microscopy 的 examination to see 这个 actually 确实是没有这个细胞在里面的。所以呢to a large extent 这里就压根就没有细胞，就是to a large extent 这个当然这个在 clinical setting 里面总是有各种各样的 noise 和 variation 的，是吧？这个是所有的 clinical setting 都会有的，但是most of the time 这个细胞是一开始就拿掉了，所以 cellular background 并没有。
好的，谢谢钟老师。那么下一个问题 from Rolling， question is nice talk. I'm wondering if it is possible the results of specific exRNA expression on AD patients can be confounded by something, for example, the drug the patients take or something comorbidity.
这是个很重要的问题，这里有2个问题。第一个，先说这个有没有其他的 confounding 的因素，可能的 confounding 因素，肯定是有的。第一 ，AD 只是人的表征的众多表征的之一，而且是一个相对来说不清楚的表征。我们刚才看了什么是 AD 啊，这个比如说开始 confuse about time 啊，这个不记事儿啊，我觉得我现在也不记事儿，是吧？这个你把人分成这个 clinical 的 symptom 的 AD 和不 AD， 这个区分是非常粗糙的，都没有什么好的 quantification 可用。但是你想人，你有多少的区分式啊？就是从他的各种老人不可能，就说我不多发点有些发炎，我们有各种各样的问题，对不对？各种 tissue 各种 organ 都有各种各样的区别，所以你就这么一分，是很多的区分法的之一。血是什么呢？血 to a large extent 它既是 provide nutrient 的 source， 但是它也是 dump 各种 waste 的 solid sewage 啊，所以各个 organ 它把这个不需要的东西不是都得 dump 到血里面去嘛？不管是我的肝还是我的肾，对不对？所以当我们想象的我在区分 AD 和不 AD 的时候，其实可能这些人他还有肾的问题，那些人他的肝已经开始不一样了，那些人他早就有 cardiovascular predisposition， 所以这些东西都是 potential 的confounding 的因素，都 potential 的需要考虑的有些 genotype 也需要考虑。另外一个呢，就是我们现在已经有很多的新的证据在男女的区别。这个男女的区别就是包括这个 PHDH 在男和女他们在早期 AD 的时候都不是说这个同时早，这个似乎男性就更早，就是更早这个 PHD 就上来了，这个是为什么呢？当然还得再研究。Comorbidity 呢指的是其他的并发症，这个 AD 常见的并发症有帕金森啊，有其他的prefrontal dementia。这个有一些其他的并发症，很神奇的是哈，这个经常我们前面的研究，经常有的时候觉得我找到了一个 biomarker， 但是呢，其实它可能不是一个 AD 的 necessary biomarker， 因为 neural degeneration， 你想 neuron 坏掉了，那会有多少问题呢，对吧？所以呢，前面经常有一些 excitement， 结果后来可能没有变成 biomarker 的一个主要的 drawback 就是它其实并不是 AD specific。就是我 neuron 出问题 ，neuron degeneration generally 都会出现的一些现象。在我们的2020年 current biology paper 里面，我们开始 demonstrate 就是 between 帕金森和 AD 之间，结果就是我们能够看到，就是说，当你只有帕金森的 至少从 brain protein level 来说 ，PHDA 低，有帕金森和 AD comorbidity， 就是这2种病同发的，它这个 population level 就跑到了中间。干净的只有 AD 没有帕金森的 population 呢，我又上一层这里我们开始这个有越来越多的证据，包括前面我讲的 publish 证据和现在更多的证据，开始这个发现这个 PHDH 似乎是和这个 specific 的这个病理有关的。当然，我们现在也开始有一些preliminary data about 这个为什么它是一个治病嘛，不是治疗的，这是 cause 病的这个原理的所以第一 comorbidity 必须被考虑，对吧？这个而且我刚才已经说了，不光被考虑，还得重要，这个事情非常重要的。它是一个 AD biomarker 还是一个 neural degeneration 的一个东西？这个就是靠 differentiate comorbidity 去 test 的。第二呢，这个劲似乎还真的是有很强的 specificity to the disease.
Okay, thank you, Dr. Zhong. Next question from Hao Yang. Yes, thanks for this very interesting talk. I was wondering if cellular PHGDH level is also enhanced in organs other than brain, especially within blood cells.
对，这个 PHGDH 在最近7年开始被 heavily pursued as a target for oncology treatment。这个原因是因为最近10年吧，这个作为 oncology treatment 最 treatment 的 research， 最近7年，最近10年人们非常 reproducible 的发现，在 cancer become progressive 的时候 ，PHGDH level 明显上调。这里其实有原因的这个就是因为 cancer 也需要最重要的这个原料，这个丝氨酸是吧 ，serine， 这个是 among most essential 的这个原料之一这个所以cancer 也要它，这个是没有问题的。如果是 cancer 要 progress into brain 的话，必须把这个上调了，因为 brain 本来就缺这个东西 in general 哈，这个我们刚才说它多的时候那个是另外一个人，但是 in general 它并不是一个丰富资源，对吧？所以这个是为什么 PHGDH 在其他的 tissue 是表达，而且在其他的 tissue 看起来不一定是它变癌，不一定是比如说 convert 成癌，而是它变成 progressive 的癌的时候，似乎这个劲就更高的高表达。所以在最近7年时间去 try to develop 这个 inhibitor of 这个 protein 的话，已经变成了一个 one of the important pursuit 但是至于它为什么就能够让癌更 progressive， 这也是一个很重要的 research direction。这个 some of the most notable journals recently 也 heavily published on 那个 front， 它其实有很 interesting 的 mechanism， 它得很会调它，才能最 progress， 才能变成最坏的癌。因为它是 essential gene， 所以它是表达在各种 cell type 里面的，包括 blood cell。
好的，谢谢钟老师。Next question from 庞飞 again。因为时间有限，我就问你提的下面第一个问题哈。So since there is significant level of variation of each individual gene in both control and AD patient group, I'm wondering whether we need a panel for reliable diagnosis of AD.
这个是非常重要的问题。AD 的 diagnosis 是在不同的 clinician 之间都是有明显不一致的，就是包括在 UCSD Research Center， 就是2个 clinician 他的对 AD diagnosis 的不一致的率都不低的，就是可以，因为这个原因还是我刚才其实已经 allude to 了，它不像 cancer， 我经常有一个客观的东西，对吧？breast cancer 不光有客观的，我还可以用客观的东西给它分亚类，对吧？但是 AD 是没有的，你的 measurement 都没有客观的东西，对吧？没有 brain biopsy， 我刚才已经说了这些了。所以这个在这个，所以我如果只拿 AD 和所谓的 AD clinical defined AD 和 control 的话，这个东西其实是非常非常不干净的这个里面我刚才就说，你换一个 clinician， 你define 的 AD 都不一样，哪些人是 AD， 哪些人不是 AD 都不一样。所以在我们的第一个 study 里面，我非常 appreciate 我的这个 collaborator Dr. Edward Cool， 他一开始就说这个东西我们只用 subset， 他做了15年的 follow up， 他说我只用那些有 brain pathology 的，就是这些人去世以后 brain 是 available 的这个就算在我们的 clinic 里面，这个也是很少的一部分人，因为他得这个满足很强的条件，他的 brain 还得 available， 对吧？然后这些 brain 以后，我做了 pathology 以后，我可以清楚的这个 rate 它是不是 AD 或者是在哪个 stage， 是吧？就是所以在我们的第一篇文章里面的 effort 全部都是AD confirmed， 就是我说的 control， 它不光是 clinical defined， 而是 pathology defined。我说的 AD 都是，我说的 converter 也都是所以这个是所以这个 panel 是必须的，也有的。当然，当我们的结果被发表以后，当然就是在各个地方都开始有各种 interest 去 follow up。这个不光在 blood， 而且在 cerebral fluid， 大家都有 interest。我们自己在这个 San Diego， 这个也有上千人的 follow up。这些 follow up 就不是每一个人都 都有这个 brain pathology data 了。Many of them are still in our clinic 这里 some of them 我们有这个 clinical diagnosis， 这个时候我们就可以开始 prospectively dissect 这个 to what extent 我们这个东西和 clinician 的这个 defined diagnosis 的区别了。当然有一些人已经过世了，那这个时候我们又可以再从他们的 brain 里面去说正确的是什么，至少从 pathology 的角度正确的是什么。这个我们已经看到了这个 preliminary data， 它这个确实发现就是说在这个血还是人活着的时候抽的血，这个血里面的这个 quantification， 对于 pH quantification 就是在和clinician diagnosis 有区别的时候，它 pathology 更支持这个血里面的 pH，所以在这个是我回答你的这个需不需要 patient defined absolutely correct AD group， 当然是需要的。但是现在我们因为在做 follow up 的 validation 别的地方也是 follow up validation， 他们就甚至可以 interrogate 我有没有这个真正的 pathology 对我结果的影响。
好的，谢谢钟老师。那么由于时间关系呢，我们再请最后一位同学来问问题。我看到开华同学你有举手，你想说一下你的问题吗？
好的，谢谢。老师你好，就是 very impressive presentation。然后我只有2个很小的问题。第一个问题是我看老师在 AD 的 gene 里边没有看到 ApoE， 您是当时刻意的，就是没有想要去查找这个 gene， 还是说就是刻意的想要另辟蹊径查找一个新的 signaling pathway？ 然后，对，我那个是 AMP AD gene 那边。
好的，对，这个 ApoE 是在 sporadic AD 里面，在 genetics risk 最强的。这个 AD 呢，就是 AD patient 里面呢，大概有 5% 呢，我们把它叫成 familial AD， 他们基本上来说必须有 mutation of one of 3 genes。这些有这3个 genes 里面有 mutation 的这些人呢 ，unfortunately 他们 usually 在 young adult， 他们不用等到65岁，他们就会有 AD 的现象。这一部分呢是大概占 5%， 剩下的 95% 呢，我们一般来说叫做 sporadic AD 自发性 AD， 他们并没有这3个 genes 里面并没有 mutation 的。我们再说一下，我把它 translate 成老鼠模型。我们也听说过有 AD Alzheimer 老鼠模型，老鼠模型全部是 familial AD， 它必须在这几个 genes 必须是动，不动的话，这个老鼠就活了1-2年，对不对？这个它是没法出 AD 这个现象的，除非你用很毒的东西去毒脑子，那个是不是 AD？ 那个其实不一定，对吧？就是，所以那么没有除了那3个最 familial AD 的那3个 gene 以外，其他的 AD 呢，就是它实际上可能都是另外一种病，对吧？那 95% 的所谓的自发性 sporadic AD 或者另外一个名字叫 late onset AD 来说，是我们 usually 说 AD 的时候，我们更多的 refer to 他们里面 so far 如果说什么 gene 是 risk factor 的话，最清楚的就是 ApoE。那么 ApoE 这个 gene 它的这个 allele 它的 genotype 和自发性 AD 的这个 frequency 是有 correlation 的。这个时候呢，首先说这个 gene most of the time 它的表象是在它的 genotype 上，不是在它的 expression 上。确实这个 gene 呢，确实在 extracellular RNA level 还确实有变化不过仍然来说，这个东西它的相关性是在它的 expression level。还是在它的 mutation 上，现在更主要的证据还是在 mutation 上。我们虽然也看到了变化，甚至我刚才没有讲，这个 ApoE 是 one of the 这个 lipid metabolism genes。实际上，你看所有的 lipid metabolism 加在一块，包括 ApoE 了。你看这里也有一个 AD 的上升的小峰，这个 indicate 就是 lipid metabolism 可能有一组 gene 呢，在 AD 里面就是有毛病的，包括 ApoE 哈。这个跟 ApoE 出毛病也是有关系的，这一组gene是也有 expression 的关系的。这一组gene有可能在 blood 里面也是有表象的，只不过是当我们在找单个的gene的时候，还不是最强的。你看，可能这里没准有一组是相关的。
对，然后还有一个很小的问题是，老师您在那个 Converse 那边的数据，它其中底下的 bottom level 那边有3个人，他的尽管有上升，其实他的那个PHGDH 的这个 not express the gene level is actually very low。对，上一张就是这几个，后边这3个，所以我在想说，是因为就您有查看，因为他这个您当时是说他有上升，跟他在发病之前的时候是有上升，那您有查过其他的 control group， 他在这些15年之内有上升吗？还是说他只是在一直很平的一个 level？
对，这个问题和前面张鹏飞的一个问题有相关性。我确实是当时为了时间，我确实是没有 elaborate 在这个事情上。这里我们有3个组哈 ，control 是放在这个蓝色的这一组了，AD 就是一开始一进来，2000年一进来它就已经是 AD 了，然后15年的 AD 是吧？这个和 converter， 你看哈，这个时候我们每一个点呢就是一个斜率了，不是一个 baseline level。baseline level 呢，我们现在还不知道该怎么用。这个 baseline level 这个跟人可能有这个 personal variation， 可能还需要再去adjust 才知道 baseline level 我能不能用吧。但是这个 slope 呢，就像是我 PCR 自己有 control 一样，对吧？因为自己和自己比嘛，我前面的就是我的 control。如果它 baseline level 人和人之间怎么比，这个对这个还是应该再 study 啊。不过在这里呢，我们先就取了巧，就没有用 baseline level， 因为可能 baseline level 跟它的变化也不是最相关，就相当于我自己有一个 internal control， 每个人都有自己的 internal control， 它的信号就出来了。就像你说的，确实是需要和 control 比的。这些 control 的人也跟了4、5年啊，他们也是同样的65岁一样的，他们也是 age match 的 sex match 的，他们在这15年里面就没有明显的变化。这里实际上每一个人我都把他的 slope 画出来了，这里面我没有去说最后有一个人可能有变化，其他的我就看他的这个 mean 加上 mean 的 standard deviation 都和0有相交的，就是这15个人这些 control 里面除了有一个人可能有上升以外，其他的这个他们跟了15年就没有变化。但是你看 AD 这一组啊，这些人是一开始就 AD 了，他的这个 PHD level 就是上下乱变，有的使劲往上，有的就往下，这个跟 converter 也不一样。这件事情我们不理解，但是呢，我们是有猜测的，就是说这些人的 treatment 是什么，不是可能是有关是吧？这个他们的 treatment 的 course 我们并没有这个 privilege 去 access 的，就是这些人他们现在的 treatment regime 就我们的 IRB 来说，并不能知道这个人的 treatment regime 但是不排除就是说这是为什么 AD 了以后，你已经是判成 AD 以后，它就变得就有各种各样的变化，这个跟他现在是怎么情况也是有关系的。但是这一组人就是至少我的 diagnosis 和他的这个基本上就是在这 time window 之间，它前后就7年左右吧，7到10年吧，就是这个 time window 是被 capture 的时候，这个时候他们似乎是 uniform 的上去的。
好的 ，OK， 谢谢老师。
没问题，好问题。
好的，那谢谢钟老师。那运行你能 share 一下 screen 吗？钟老师可能你得先 stop share， 对。好，那么在我们最后讲座的最后呢，这是我们社群小助手的微信，大家可以还没加入社群的朋友们可以扫码，然后添加小助手就可以入群了，只需要实名验证，其他没有任何附加条件。然后我们的社群目前暂时未对本科或者硕士研究生群体开放，但我们的公开活动欢迎各位的参与。最后呢，我们近期在收集大家对于邮箱订阅的倾向，所以最后这个2维码呢，就是关于邮箱订阅的，只需要填写第2题和第7题就好了。然后感谢大家对我们的帮助。那么最后呢，再次非常感谢钟老师给我们这次amazing talk， 非常的
谢谢大家 share Sunday with me. Some of you are Monday morning, early Monday morning with me. 谢谢组织者冯源，
是的，然后
运行，
对 对，并且非常感谢各位同学的参加，辛苦你们了。好的，那本次学术讲座就到此结束，谢谢大家。那钟老师您稍微留一下好吗？
OK。